Hi, good afternoon. I've been informed that the reception, which follows the Walls lecture, will be held in the lobby of the entrance to the clinical center behind me. There's some construction or something going on in the usual place. So you can get there by going out either of these two doors. So this is the first Walls lecture of the new series, and I am personally extremely happy to know that it's going to be given by Helen Piwnica-Worms, whom I would like very briefly to give you a little background information about for those who don't know her or her outstanding work. Helen is the Gertie T. Corey Professor of Cell Biology and Physiology, and also Professor of Internal Medicine at Washington University School of Medicine, and also a Howard Hughes investigator. She received her PhD from Duke in microbiology and immunology, and then took a postdoctoral fellowship with Tom Roberts at the Dana-Farber in Boston, where she was studying protein tyrosine phosphorylation in relationship to cell growth and transformation. At the conclusion of her fellowship, we were very fortunate to recruit her to the department, which I chaired at the time, in physiology at Tufts. Within a short period of time, a matter of, I think about 4 years, or maybe it was 5, several things happened. One, she quickly established herself as an outstanding mentor, attracting a group of very bright students, fellows, colleagues, and really made remarkable progress in her research, and also in cooperating with everybody with regard to the both educational and even political aspects of the department. During that time, she spent a part of a summer at the marine biology lab at Woods Hole, where she became interested in the then emerging field of the molecular biology of cell cycle. And bravely, I guess that's the right word, she entered a field where there was a large number of very heavy hitters around the world who were beginning to relate the specific steps in the eukaryotic cell cycle to specific phosphorylation of different proteins and so forth. But she made her mark, and so successfully that she was then recruited back to Harvard Medical School, where she continued this work, continuing to make outstanding progress and receiving many awards along the line, and then was recruited to Washington University School of Medicine, where she has been ever since. Her research has concerned the regulation of the eukaryotic cell cycle, particularly how entry into mitosis is regulated, and how cells are delayed in this process, so-called checkpoint control, which is the subject of her seminar today. As I mentioned, Helen is a Howard Hughes investigator. She's a member of numerous scientific advisory boards, including the American Association for Cancer Research, and she was elected as a fellow of the American Academy of Arts and Sciences. She's also the senior editor of the journal Molecular Cancer Research. I think the success of an individual in science is determined by, in large part, the people who are mentored. And Helen has had an extraordinary record and now has mentored over 30 trainees since becoming an independent faculty member back when. She's very active in the MST program, and she's played a critical role in the building of a molecular oncology program at WashU, which links the Department of Cell Biology and Physiology, the Department of Medicine, and the Cancer Center. I find it exciting that Helen is the Gertie T. Corey Professor, because many, perhaps in this room, who don't have gray hair or no hair may not know or remember who Gertie T. Corey was. She and her husband Carl won the Nobel Prize in 1947 for their remarkable, important work in the broad field of metabolism. She was one of the first women to break the so-called glass barrier, and I think it's entirely appropriate that today we're going to be honored by having another extremely talented scientist who also carries that designation as the Gertie Corey Professor of Cell Biology. So Helen, it's with great pleasure that we welcome you back again to NIH, and the title of her seminar is up there, or will be shortly, the control of checkpoint control in relationship to breast cancer. Welcome.
Thank you. Well, it's my great pleasure to be back at the NIH, and particularly exciting to have Wynn invite me. As he mentioned, he was the person responsible for first launching my career, and he's been a wonderful mentor and supporter throughout my entire career, so thank you, Wynn. So I would like to describe to you today some of the pathways that we've been studying to understand how the cell division cycle is regulated, and then how cells stop in response to various stresses. And then I'd like to discuss how we've taken this information now to build some preclinical models and have actually taken some of this work into clinical trials. So I'm going to be focusing a lot on this protein today. It's the CDC25a protein phosphatase. It is a positive regulator of the cell division cycle. And the way it operates is to dephosphorylate the cyclin-dependent protein kinases. So it's part of the cell cycle machinery that is important for driving both early and late cell cycle transitions. Now in mammals there are 3 members of this family, and we'll come back to that later. So it ends up that this phosphatase is highly labile in cells. It has a half-life of less than 30 minutes. And when cells are exposed to stress, this can be in the form of replication stress or DNA damage, within 30 minutes all of this phosphatase is gone from the cell. And the idea is that by eliminating this positive regulator of the cell cycle, it gives the cell an opportunity to pause and correct any damage before proceeding on through the next transition. Now, failure to degrade the phosphatase in the presence of genotoxic stress causes loss of 2 important checkpoints in the cell, both the S and G2 checkpoints. And this has been observed to cause preferential killing of P53 deficient tumor cells. And so this will provide the rationale for the clinical trials that we're running. So in terms of cancer, you frequently find this phosphatase overproduced, and when you do, it correlates with poor prognosis. The mechanisms leading to its overproduction in these various tumors is largely unknown. And so pharmaceutical companies have actually been devising ways to perturb this phosphatase in a couple of ways. In one case is to develop inhibitors to block the cell division cycle. And the second is to actually develop ways that prevent the phosphatase from being degraded in response to either chemotherapeutic agents or ionizing radiation, to then force these P53 deficient cancers through the cell cycle. And so the way that's being accomplished is by developing inhibitors against Chk1, and again, that forms the basis for the clinical trials. So the phosphatase is highest in mitosis, and as cells exit mitosis, it's degraded, and that occurs through the APC E3 ligase, and the targeting subunit here is CDH1. A transcriptional program ensues in G1, and the cell starts to accumulate the phosphatase, but its accumulation is counterbalanced by another E3 ligase, beta-TRCP. And so that's operating throughout interphase to maintain appropriate levels of this phosphatase in cells. And it ends up, there's an important regulatory region within the amino terminus of the phosphatase that is responsible for mediating the targeting of this E3 ligase to CDC25A. And it requires phosphorylation of a serine residue, serine 82, to create this DSG motif, which brings in beta-TRCP to degrade the phosphatase. And so we've been working out mechanisms to understand how this, what other phosphorylation sites within this region are important. And what we've published last year is that in early cell cycle phases, GSK3 beta phosphorylates serine 76 within the amino terminus. Now, GSK3 beta often requires a priming phosphorylation site, and that function is served by phosphorylation of threonine 80, and that can be catalyzed by PLK3. Now, as cells proceed through the cell cycle, Chk1 then takes over during the S and G2 phases, and it phosphorylates serine 76, and it does not require a priming phosphorylation site. But what then one finds is a hierarchical phosphorylation that occurs, with serine 79 occurring next, serine 82 then following, bringing in beta TRCP and ubiquitin-mediated proteolysis. And so as we've been able to discover some of the pathways that are being regulated here, we are constantly asking whether we can now go into cancer cells and figure out if any of these pathways we've discovered are perturbed in these cells. And so one can imagine this could occur through the CDC25a regulatory kinases, the E3 ligases themselves, deubiquitinating enzymes. Some cancers might even contain mutations within this the gene encoding the phosphatase, or microRNAs. And so I just want to describe one pathway we've identified that may explain some of the ways that CDC25 is overproduced in these tumors. And so in this experiment, 142 tumors were examined on a tissue microarray. And what we looked at was how many of these tumors have inactive GSK3 beta, and we can look at that by the ability of a phosphospecific antibody that recognizes the inactive form to stain these samples, and then how many also overproduce CDC25A. And so this is summarized here. Out of 142 tumor samples, 42 had inactive GSK3 beta, 44, and 42 of those overproduced CDC25A. So that's a pretty significant correlation. Now, interestingly, 98 of the tumors had active GSK3 beta, and 61 of those also overproduced CDC25A. So although it suggests GSK3 beta inactivation may account for a subset of the overproduction in CDC25A in these tumors, we still have a lot to learn, and so we're following up on this now. But for the rest of my talk, I want to talk about how Chk1 and CDC25A interact. And so there are at least two ways that Chk1 regulates this phosphatase. As I mentioned, the first case, it phosphorylates serine 76, and that then serves as an important first step in getting this phosphatase to be degraded. Now, there's a second phosphorylation site within the C-terminus at threonine 507. And it ends up that within the C-terminus of the phosphatase, there's a docking site for CDK1. So CDK1 is really one of these master regulators of mitosis. It must be activated to drive cells into mitosis. And so you really do not want CDC25A to activate CDK1 at inappropriate times in the cell cycle. And one of the ways you prevent that is by docking 1433 proteins to basically keep the site blocked from CDK1 docking. Okay, so at least two mechanisms then for this form of regulation. Now, Chk1 itself then is subject to many forms of regulation. And one of the major pathways is through replication stress. So in the presence of replication stress, the ATR protein kinase becomes activated. This in turn activates Chk1 to ultimately cause CDC25A to be removed from the cell. Now, a few years ago, we discovered a feedback pathway involving PP2A. So it ends up that ATR is constantly phosphorylating Chk1 all the time. The system, it's constantly occurring. But if you look in cells that have not, where you've not really artificially induced any stress, you really don't see Chk phosphorylation. And that's because there's a feed forward and feedback pathway that involves the PP2A phosphatase. So PP2A is constantly dephosphorylating Chk1 to keep it in sort of an inactive state. And we propose that you keep this pathway on so that cells are poised to respond very quickly to either replication or genotoxic stress. Now, there are two E3 ligases that actually target Chk1. So Chk1 is itself ubiquitinated. We discovered the Cul4 DBB1 pathway, and more recently there was a paper published by Tony Hunter's laboratory showing that the Cul1 FBX complex degrades Chk1. And so, also HSP90 is required to make active Chk1 in vivo. And so there are inhibitors to various forms of these pathways, and I'm going to be coming back to some of these Chk1 inhibitors that can inhibit Chk1 and thereby perturb the Chk1cdc25a pathway. Now, in conjunction, there's another stress pathway that responds to DNA double-strand breaks. That activates the ATM protein kinase, and that then is important for feeding into the P53 pathway. And in response to these various forms of damage, cells can either elect to undergo cell death, or they can elect to stop at these checkpoints and try to repair some of this damage. And so we're going to be discussing how we can utilize this pathway to preferentially kill cancer cells that lack this pathway. So what's then the rationale for the trial? Well, several years ago, it was demonstrated that genotoxic and replicative stress will basically induce cell cycle arrest. But if a cancer cell lacks P53, it's unable to arrest in the G1 phase of the cell cycle. So it can't arrest here, and there you remove this whole P53 mediated form of apoptosis. Now, although cells can stop in the S and G2 checkpoints because they have the Chk1 CDC25A pathway, they are somewhat weakened because P53 reinforces these pathways. And so what Art Pardee showed in the mid-1980s was that if you added a compound like caffeine, and then later we, in collaboration with Patrick O'Connor and Ed Sosville, looked at targets of UCN01. Basically what happens now is you also lose the S and G2 checkpoints. So a P53 deficient cell will lose all these checkpoints, whereas a P53 proficient cell can initiate these two but lacks this one. And again, you see this preferential killing of P53 deficient cells. So it was later shown by Claire McGowan that caffeine inhibits ATR and ATM. And like I said, collaborators at the NCI, who were then at the NCI, Pat O'Connor and Ed Sosville, in conjunction with my laboratory, showed that UCN01 is a very potent Chk1 inhibitor. So we know the targets. So the rationale for the trial then is to treat patients with a DNA damaging agent. And in our case, we're going to be using irinotecan, a topoisomerase 1 inhibitor. And one would expect CDC25A levels to fall. We've shown this in many experimental models. You then come in with a Chk abrogator, in a Chk1 inhibitor, UCN01. And I just want to point out that Chk1 it inhibits a lot of protein kinases, okay? But you can completely mimic the effects on the checkpoints by using Chk1 siRNAs. So we think that the property of UCN01 inhibiting Chk1 is responsible for the checkpoint abrogation we observe in our system. So what happens then is A levels rise, and instead of stopping in the cell cycle, you blast through those checkpoints, and we again see this preferential killing of P53 deficient cells, which also lack the G1 checkpoint. And again, UCN01 is not a specific inhibitor. In addition to many other kinases, another enzyme it inhibits quite potently is PDK1. And this is going to be important again because you affect the AKT pathway with UCN01. So we'll come back to that. So the first phase 1 clinical trial that we performed was done in collaboration with Paula Fracasso, who is now at UVA, Howard McLeod. So Paula was our oncologist. Howard McLeod is our pharmacogenomics expert. He's now at Chapel Hill. And so the recent studies are done in collaboration with Cynthia Ma and Matthew Ellis, who are in our oncology division at WashU, and they're both breast oncologists. So in the first trial, 25 patients with metastatic cancer were treated, again, with irinotecan followed by UCN01. Patients had a variety of solid tumors. 60% of those patients exhibited a response on this clinical trial. And it ends up that 10 of our patients were breast cancer patients, and we observed something interesting with a subset of those patients. So 4 of the patients had triple-negative breast cancer, and that's a form of breast cancer that lacks estrogen receptor, progesterone receptor, and does not overproduce HER2/neu. And it's a form of breast cancer where there really are no targeted therapies. And it ends up that 4 out of those 4 patients responded, 2 with a partial response and 2 with stable disease. And what was remarkable is if 2 of the patients who had metastatic lesions to their chest wall and lymph nodes responded really quite remarkably. Within the first cycle of treatment, you basically saw complete loss of the tumor on the chest wall. And so based on these results, the NCI has agreed to let us extend this trial, and we're specifically enrolling patients with triple-negative breast cancer. Now, in thinking about why this particular subtype might have responded to this therapy, we realized that P53 mutations are actually common in breast tumors, and in particular, triple-negative, at least, this is probably a low estimate of 44% one finds mutations in P53. And in the other forms of breast cancer, so for example, with the ER positives or HER2 positives where there are targeted therapy, often the tumors that don't respond to those targeted therapies are ones that also have mutations in P53. So we wanted to then, with this first trial, do some correlative studies. And as I mentioned, another property of triple-negative breast cancers is you frequently see that they silence PTEN. And so we were wondering, well, perhaps the reason UCN01 is working so well is that you have a P53 mutation as well as activation of the AKT pathway. And the non-specificity of this inhibitor is actually is a plus in this situation. And so on this first study, we were only able to get 2 biopsies from the actual tumor itself. And I'll take you through what we've been able to do with that. But in all cases, we were able to get blood. And the reason the correlative studies with UCN01 are so important is that it's been shown to have, first by here at the NCI, to have a very long half-life in patients. And we confirmed that in our study. So it leads one to wonder if UCN01 is even bioavailable at the concentrations used in these trials. And so to look in the blood samples as well as the biopsy samples, we're going to use phospho-S6 as a readout for the ability of UCN01 to inhibit PDK1 and then the AKT pathway. So here's a biopsy sample, and we were only able to get at baseline, and then the patient would come in and get irinotecan. 24 hours later, UCN01, and 24 hours later come back for this biopsy sample. Now, this particular patient actually was one of the patients with triple-negative breast cancer that responded on the trial. We were able to get sequence on her tumor. It was mutant for P53. And you can see a nice inhibition of phospho-S6 with UCN01 in the tumor. So basically, we've had pathologists score all these samples, and phospho-S6 was observed in about 40% of the cells in the pre-therapy sample, and that was reduced to about 5% in the post-therapy sample. The second biopsy sample came from a patient that did not respond on the trial, also with triple-negative breast cancer, and this patient actually, sequencing of the tumor revealed it was wild type for P53. So it's a very small cohort. I don't want you to walk out with any conclusions, but just it's intriguing that we would predict a P53 mutant might respond, whereas a P53 wild type might not. But again, in both cases, we see a significant decrease in phospho-S6 staining in the biopsy samples. Now, again, we were able to get blood from the patients, and this was nice because you can get blood pre-therapy, after 24 hours after irinotecan, 24 hours after UCN01, and then the patients come back in on day 8 for another irinotecan treatment. And you can see with phospho-S6, with the UCN01 treatment, a nice decrease here. And basically, by compiling all the data, you can see on day 3 a nice decrease in phospho-S6 staining. Now, you'll notice some rebounding on day 8. And because the half-life of UCN01 is so long in patients, they come back for 2 irinotecan treatments before they even get their second UCN01 treatment. And what this might be telling us is that it's losing its bioavailability by day 8. And so the scheduling here might not be quite optimal. So in conclusion then, we observed partial responses in 2 patients with triple-negative breast cancer, and sequencing of DNA in one of these tumors demonstrated a P53 mutation. We saw reduced phosphorylation of phospho-S6 in blood, normal rectal mucosa, which I didn't show you, and tumor biopsies 24 hours post-UCN01 treatment. We confirmed the long half-life of UCN01, and as I mentioned, we have the extension phase of this trial going on right now. And in the extension phase, patients are being treated at the maximum tolerated dose. We're trying to obtain as many serial biopsies as possible to assess the important pathways that need to be assessed. And we also now have written a protocol for combining gemcitabine with a more selective Chk1 inhibitor. This is an AstraZeneca drug, and so this will be the next trial that we hope to start within the next year. Now, I realize this was my first foray into clinical medicine, and I understand now how hard it is to do clinical translational work, okay? So trying to get the appropriate samples so one can really do the correlative studies to really come back and try to test the hypothesis you're trying to test is very, very difficult. And so we wanted a way to develop a system that we could bring back into the laboratory to begin to ask the types of questions we're used to asking under controlled situations. And so I'm going to describe a preclinical model that we've set up. This was first described by Charlotte Cooper-Wasser, and our collaborators, again, include my colleagues Matthew Ellis and Cynthia Ma. Rebecca Aft is a surgeon at WashU that gives us fresh biopsy samples, and we also obtain samples from the Cooperative Human Tissue Network. And so the idea here is to take fresh biopsy samples from the patients and then engraft them into the humanized mammary fat pad of non-skid mice. So for those of you who aren't familiar with this, basically you take immunocompromised mice, non-skid mice, you clear out the 4th mammary gland, and then you come in and you humanize them with fibroblasts. And these fibroblasts were derived from a reduction mammoplasty. They're immortalized by expressing telomerase, and they also express GFP. So you let the tumors engraft and the mice recover, and then you come in with fresh biopsy samples that you basically mince so that they're in little organoids. And you transplant those, and once they take, it's tricky to get this to work, but once they take, we're able to passage them ad infinitum. So we're then able to obtain enough mice to do what we call our preclinical trials, and then hopefully be able to do survival studies. So here's just an example of what the tumors look like. We know they're human because we can stain them with human-specific cytokeratin. And we've been able to establish 10 models now. Some are from primary tumors, some are from metastatic lesions. Interestingly, tumor 5 is a brain met from the patient that came in here that had a primary tumor initially. And so some of these are triple negative, some are ER positive. We have one HER2 overproducer. And we know that P53, we've sequenced P53 in all of these tumors, so we know whether they're P53 mutant or wild type. So I'm going to show you some of the preclinical mini trials that we've put the mice on. And basically, what we do is we take a cohort of mice that have been engrafted with these tumors, and we either treat them with vehicle, with the DNA damaging agent, the Chk inhibitor, or the combination. This is a nice control. You can't do this in the patient, this control alone. And then we isolate the tumor, and we either look by Western blotting or immunohistochemistry at the pathways we want to interrogate. And these are some of the pathways we interrogate. So when you treat with irinotecan, it'll induce single-strand breaks that are then converted to double-strand breaks, and we can monitor that with gamma H2AX staining. Irinotecan should induce arrests in both the S and G2 phases of the cell cycle, and geminin is a marker for those cell cycle phases. We look to see how cells are responding. Are they blocking before mitosis? And if so, we should see reduced phosphohistone H3 staining. And if you bypass the checkpoint, now you should see phosphohistone H3 staining go up in the presence of DNA damage. And then finally, we ask is apoptosis activated, and we do this by looking at cleaved caspase 3. And then we look at the AKT pathway again by using phospho-S6. So here are three of the tumors. HIM2 is a mutant, HIM3 is wild type, and HIM4 is mutant for P53. Here we've stained for P53. Whenever you see high staining, you know it's a mutant. And here's phospho-S6. So you see each of these tumors have various levels of phospho-S6 staining. Now, just because a tumor sequences wild type for P53, it doesn't mean the pathway is intact. And for our studies, it's really important for us to know this. And so we treat the mice with either vehicle or the DNA damaging agent, and we ask, is P53 stabilized, and is its downstream target turned on, and does it accumulate? So in this case, P21. So in HIM3, which is sequences for wild type P53, you see P53 accumulate and P21 accumulate. So we label this as a functional P53 pathway. These two models, you see here, HIM4 that's staying very high for P53. You see high basal levels of P53 that really don't change in response to damage, and no real reproducible effects on P21 levels. And then this is a mutant that makes a truncation, so we don't see the protein made in the cells. All right, so here's a case where we've treated each of these mice harboring each of these tumors with either vehicle, irinotecan, UCN01, or the combination. And we're looking at phospho-S6. Now, all of this has been quantitated, but I'm going to show you the visual pictures. You can see UCN01, nice decrease in phospho-S6 relative to the control, suggesting that the AKT pathway is being inhibited during treatment, and that stays low with the combination therapy. Here's the HIM5 model, which was a mutant model, and here we're staining with various markers that I described to you. And so at baseline, if you look at geminin, you see a significant amount of S and G2 cells that goes up when we treat with irinotecan, which is what we'd expect because these cells should arrest in the S and G2 phases of the cell division cycle. So we expect that to go up. With UCN01, you'll see, we have quantitated this. There was really no significant difference from baseline, and with the combination, a little higher. Now, if you look at phosphohistone H3, you see, again, it's a mitotic marker, a certain number of mitotic cells. When you treat with irinotecan, this goes down as expected because you should be delaying entry into mitosis. UCN01, really no difference from baseline, but now you can see with the combination therapy, a significant increase in the number of mitotic cells, suggesting a checkpoint bypass. With gamma H2AX, this tells you how much double-strand DNA breaks you have, and again, you see it increase with the DNA damaging agent. You can even see a further increase with the combination therapy. Looking at apoptosis, again, with the DNA damaging agent, a little bit, but a much higher increase with the combination. So the markers are responding in the way we'd expect them to respond. Now here we've looked at gamma H2AX just to show you that whether you're P53 wild type or null, they basically, in response to irinotecan, generate similar amounts of DNA damage as indicated by gamma H2AX staining. But only in the P53 mutants do you see this synergy of the combination therapy, where you see much higher levels of gamma H2AX staining. Now we can also look at Western blotting. So this is just combining from the HIM3, the wild type P53 tumor with the two mutants. And again, you see the combination therapy, and even irinotecan alone is able to induce apoptosis. And you see much less in this P53 plus model, again, with the same with immunohistochemistry. So again, we've quantitated all this. I just want to bring your attention to a couple of the panels. So with phosphohistone H3, you'll note that in a P53 wild type tumor, you see a decrease, which we'd expect because checkpoint should be intact and cells should be delaying entry into mitosis. But you can see the two mutants we see with the combination therapy an increase, suggesting that they have bypassed this G2 checkpoint and moved into mitosis. With cleaved caspase 3, again, you see only in the P53 mutants do we see this induced apoptosis in response to the combination therapy. So UCN01 in this model we've demonstrated decreases phospho-S6 levels, whether you're wild type for P53 or mutant for P53. However, where you begin to see the difference is when you look at cell cycle arrest abrogation So you see UCN01 abrogates cell cycle arrest induced by irinotecan and induces apoptosis in mutant, but not wild-type P53 triple-negative breast cancers. So what do we see as the promise of this model? Well, we can get sufficient amounts of material to do all the type of genomic and expression type experiments that one might wish to do. We can run properly controlled preclinical trials. We can now test different DNA damaging agents in combination with more selective Chk1 inhibitors, and we can change dosing and scheduling. And I'm going to show you just one experiment we've done with AstraZeneca's Chk1 inhibitor. And this inhibitor is currently in phase 1 trials with gemcitabine and irinotecan. And finally, we can obtain as much pre and post therapy tumor biopsies for all the biomarker analysis we need on using this system. So here's the one experiment I want to show you where we've looked at the AstraZeneca drug. So this is, again, a Chk1 inhibitor. It's reported to be more selective than UCN01, but my view always of these inhibitors is, it just has a different spectrum of non-specificity. So none of them are specific exactly for their target. So it inhibits other things besides Chk1, but what's nice about this particular inhibitor is we've shown it does not inhibit PDK1, so it doesn't affect the AKT pathway. So you can see with the wild-type P53, whether it's control or put on the combination therapy, there's very little apoptosis. You can see some here with the combination therapy. But again, with the P53 mutant, you see a lot of cleaved caspase 3 activation, and it might even be significant here as a single agent. Here's the phospho-S6. So you can see if you just look at irinotecan alone or AZD alone compared to the control, there's very little difference in phospho-S6 staining. So this tumor is responding to the combination therapy in the same way as AZD and UCN01, and yet in one case we're inhibiting PDK1 and the AKT pathway, and the other we're not. So it gives us a way to start segregating which kinases are really going to be important in forcing these tumors into an apoptotic cell death. So I'm going to turn now and leave that model and then describe a series of experiments we carried out to basically ask how important really is P53 status for this response. So if you look at all of the literature that has been published on showing this preferential killing of P53 deficient cells, it's all been carried out on tumor cells that we grow in the lab or we grow as xenografts in animals. And we know that these tumors have many, many mutations in addition to P53. And so we wanted a system that was basically normal except for P53 status. And we also wanted to do this in the context of just normal tissues of an animal. And so I'm going to be showing you experiments where we've looked at P53 wild type and P53 null mice, and we're going to be putting them on the combination therapy and then asking how do the small intestinal epithelial cells respond? And do we see a differential response depending on P53 status? So for those of you who aren't used to thinking about the small intestine, it is the most rapidly proliferating organ in the mouse, and all of the proliferation occurs within the crypt. This is where the stem cells are housed. They give rise to transient amplifying cells, which then go on to differentiate into the 4 epithelial cell types of the intestine. And again, we're going to be putting the mice on the therapy and then just looking at the same pathways that I've already gone over with you. All right, so let's just start by looking at gamma H2AX staining. So these are the small sections of small intestine from P53 wild type mice that have been treated with vehicle, UCN01, the DNA damaging agent, or the combination therapy. And you can see, again, the DNA damaging agent, irinotecan, induces gamma H2AX staining, as does the combination therapy. You see the same thing in the null mouse. You see staining only when you treat with irinotecan or with the combination therapy. So you get equivalent amounts of DNA damage in this model, and this is just quantitated here. So independent of P53 status, irinotecan induces similar amounts of gamma H2AX staining. So if you look now at geminin staining, so again, geminin stains S and G2 cells. About 40% of cells in the crypt are actively dividing. And so you see a high baseline staining, which can be enhanced with the DNA damaging agent. Where you start to see a difference then is the combination therapy. So cells that are wild type for P53 seem to hold this SG2 arrest, whereas you begin to lose geminin staining indicative of loss of SG2 cells in the P53 null situation. So where do they go? Well, you see a concomitant rise in the number of phosphohistone H3 staining cells, suggesting that the loss of the SG2 cells can be accounted for by the gain in the mitotic cells. So again, it suggests a checkpoint bypass selectively in P53 null mice, at least in the epithelial cells of the crypts, compared to the wild type situation. And now if you look at cleaved caspase 3, well, it ends up that, if you have wild type P53, they will undergo apoptosis with the DNA damaging agent alone. You see about, one apoptotic cell in about every 2 crypts. But you can see in the P53 null very low levels of cell death until you put them on the combination therapy, and then you can drive them forward. Okay? So now the way, if you read the literature in the field, it's been proposed that cells bypass their checkpoint, undergo a mitotic premature entry into mitosis or a mitotic-like catastrophe, and that's what then drives them into an apoptotic cell death. And so we wanted to ask how many of these cells actually stain for both phosphohistone H3 and cleaved caspase 2. And that would support the model that you must undergo this premature mitosis before undergoing death. And it ends up it's really a very small fraction. So in the crypt, so here's a P53 null mouse treated with irinotecan, and we never find a cell that co-stains. You can find cells, you see here in green, this is for cleaved caspase 3, and a red cell, which would be a mitotic cell. Now with the combination therapy, you begin to see some of these co-stained cells, but it ends up, if you really quantitate this, if you look in a P53 null mouse, 50% of the crypts contain at least one apoptotic cell, but only 1 out of 50 crypts contain an apoptotic cell that also stains positive for phosphohistone H3. So we see a lot of apoptotic cells that don't exhibit phosphohistone H3 staining, suggesting there's other mechanisms to induce apoptosis independent of this checkpoint bypass. And we also find cells that stain for phosphohistone H3 that don't stain for cleaved caspase 3. And I don't know if this is because they haven't had time yet to undergo apoptosis, or if there's other ways to just kill these cells without going into apoptosis. All right, so what this system has told us is that indeed P53 is a key determinant of how cells respond to DNA damage, followed by SG2 checkpoint abrogation. But the results we obtained were pretty minor compared to what you see in the HIM model, or if you take cells cultured in the laboratory and subject them to this treatment. And so we conclude that the mild responsive cells that are P53 null but otherwise normal, and so in this case the intestinal epithelial cells, compared with P53 deficient cancer cells grown in culture or as xenografts in mice, suggest that the additional mutations present within P53 deficient tumor cells makes them particularly sensitive to this therapy, suggesting again this might be a good way to go clinically. All right, and so I'm going to now switch to another model we developed that allows us to compare how cells respond to acute disruption of the cell division cycle compared to how they respond to DNA damage-induced cell cycle arrest. So we know that the mainstay of cancer therapy is chemotherapeutic agents or ionizing radiation, and these are DNA damaging agents. And there's a lot of side effects from this treatment, including, one of the major side effects is in the GI tract. You can get this mucositis. And so we wanted a way to ask, what if we approached this in another way and just tried to use inhibitors that would just lock down the cell cycle without inducing DNA damage first. And so I'm going to compare how the mouse intestine responds to irinotecan versus lack of cell proliferation. And we've been able to accomplish this by deleting the CDC25 family of protein phosphatases. So, as I mentioned, in mammals there are 3 members of this family. And we've been studying this family ever since I was an assistant professor at Tufts, okay? And yeast survive happily with just 1 member. And so we've always wondered why mammals have 3, and can we, functionally try to distinguish these phosphatases. And so over the years, we've been trying to disrupt these in mice. And we published several years ago, if you disrupt one of the family members, CDC25C, which was supposedly the master regulator of mitosis, we can find no phenotypes. Even if we do a partial hepatectomy and drive the liver into the cell division cycle, in the absence of CDC25C, they regenerate in an indistinguishable manner. So Peter Donovan's laboratory knocked out CDC25B, and he showed that these mice were normal, except females are sterile in this case. So then we thought these were the 2 phosphatases that were thought to be, again, the mitotic regulators. And so we said, all right, let's breed these 2 together. And again, we were able to get mice that expected Mendelian ratios. The females obviously were still sterile, but otherwise the mice are healthy. We can make MEFs from those lines. They proceed through the cell division cycle. So this then led us to actually begin focusing on CDC25A. And at the time we made this observation, CDC25A was not thought to have any mitotic role at all. So we subsequently went back to reinvestigate this question and found indeed it has a very important mitotic role. Now, if you delete CDC25A on its own, it's an embryonic lethal. The mice, you can get to the blastocyst stage, but the embryos are unable to implant, and so it's a post-implantation defect. And so in order to knock out 3 in adults, we had to go to a conditional approach, and basically we take BC knockout animals. These have to be males. We breed them to mice that have 1 floxed allele of CDC25A and 1 null allele, and then we come in with either a Rosa-Cree ERT, so this is more global deletion, or a Villain-Cree inducible transgene to specifically knock out these phosphatases in intestinal epithelial cells. And after you can imagine all the breeding it took, we eventually got to mice that basically had 1 allele of CDC25A. So mice with 1 allele of CDC25A are happy, they can live, they have normal cell cycles, et cetera, except females are still sterile. And then you can treat with OHT if it's the Rosa line, or tamoxifen if it's the Villain-Cree line. And what we found is cells. So basically, within 7 days, these animals are dead. And the reason they die is you basically completely shut down proliferation in the small intestine. So here we're looking at mitotic figures in the crypt of a small intestine. So you see mitotic figures in the wild type animal, in the BC knockout mouse, but they're absent here. If you do BRDU staining, you see a complete absence of BRDU staining. And if we started to quantitate, you might think that you'd see some apoptosis. We see no apoptosis in this model, which is really exciting because all of the models for blocking cell division in the small intestine have all induced apoptosis. So this is one of the first models where we see no cell death, just a block of cell division. And what we see is that the mitotic cells go down to none, no apoptotic cells. The Panna cells have the longest half-life of all the cell types in the intestine, so they stay approximately similar in number in this 7-day period. But the number of cryptepithelial cells diminish quite a bit. And actually, if you look at the length of the villi, they shorten significantly within this 7-week period. And this was just published, so I just want to briefly remind you, it's an interesting model in additional respects as well. So what happens in this model, when you acutely disrupt cell division in the small intestine, what happens is the cells experience this stress, and you get this compensatory upregulation of Wnt signaling. And so what we found is that Wnt signaling is high, and so every progenitor in the crypt that can differentiate starts to differentiate along their lineages. This includes cells in the base here, these CBC cells that Hans Clevers claims are stem cells. So there's two models of two sort of disagreement in the field about who the intestinal stem cell is. So there's evidence from the Clevers lab that this CBC cell is the stem cell, and then others claim that this cell in the +4 position is the stem cell. So everything begins to differentiate, this CBC differentiates, and one cell sits there and does nothing, and that's the cell in the +4 position. So we see nuclear beta-catenin within this cell, but it's not responding to Wnt signaling by differentiating. So we can't conclude whether that's a stem cell or not, but I think it's interesting that if these are a stem cell at the base of this crypt, they adopt the fate of their nearest neighbor in response to cell cycle disruption and enhanced Wnt signaling. Okay, so we see these dramatic changes, but one of the reasons we looked at Wnt signaling is because when we acutely knock out cell division, the crypts are maintained in our model. So we lose villi length, but overall the shape and the structure of the crypt is maintained. So when we compare now irinotecan treatment to the triple knockout, we see something very different. So in both cases we lose proliferation, but in the case of the DNA damaging agent, you start dropping out your crypts. So here you lose your crypts. In the case of the CDC25 model, you maintain your crypts. And this is important because this is what's housing the stem cell. In the irinotecan model, you see apoptosis, and you also see a loss of the goblet cells with an increase in inflammation, which you don't see in the triple knockout model. So by acutely disrupting cell division, you're not inducing an inflammatory response, you're maintaining your crypt. And so basically, you know, both cases you lose proliferation. With the DNA damaging agent, you get apoptosis. With the knockout, model, you get no apoptosis. In one case, you lose your crypts. In this case, you maintain your crypts. Here, you lose barrier function and get inflammation. Here, your goblet cells are intact, and you see no inflammatory response. So it ends up that, you know, cancers overproduce either CDC25A or B. We've never seen any evidence of CDC25C overproduction. So the question is, if you had a tumor that overproduced CDC25A and you had a selective inhibitor, it should be there should be really no side effects in any tissue if you can just target one of these. And so the idea is that inhibitors that block components of the cell cycle engine may be predicted to have fewer deleterious GI side effects than the current therapies that are being used. And then I'm going to just end with 3 more slides and just show you a new model that we've developed with my husband, who runs our molecular imaging center at Washington University. So, you know, most of our work has been carried out with cultured cells in a test tube, and we've come up with these beautiful models about how Chk1 works and how CDC25A is a target, and, you know, we get reproducible results about adding a DNA damaging agent, and now you come in with the Chk inhibitor and you put together these beautiful models. But the question is, at the end of the day, are these models really operating within normal cells and within the context of normal tissues and a normal vertebrate animal? And so we wanted to develop ways to interrogate these pathways non-invasively in animals, and we chose mice. And what we did was to make a knock-in mouse where we knocked downstream of the termination codon of CDC25A click beetle red luciferase. And we did this either in a form where we can constitutively make the reporter protein or conditionally. And in this case, we bred it to a villain-Cree promoter, so now you can see only in the intestine do you see bioluminescence. Now, you can say you've lopped this big protein on the end of a really important regulated protein. How do you know you haven't messed up this protein? So we made MEFs, we looked at cell cycle regulation, we measured half-life, we looked at response to DNA damage. Everything we looked at led us to conclude that the fusion protein is reflecting properties of the endogenous protein. So we feel comfortable now with this model to then begin to interrogate how this phosphatase responds to various stimuli. And so here is just showing, again, the mice minus tamoxifen plus tamoxifen. We turn it on specifically in the intestines. You can pull both the large and small intestines out, and you can see the phosphatase. And so now we said, well, let's treat these mice with irinotecan. Does CDC25A really turn over? And it ends up, within this 24-hour period, we see a destruction in bioluminescence. And if we give these mice bortezomib to inhibit the proteasome in vivo, we see A levels rise. So it looks like It's regulated inside the animal in a proteasome-specific manner, and that DNA damage is reducing its levels. Now, if we come in with UCN01, we'd expect that we should inhibit Chk1 and the phosphatase level should rise, and that's exactly what we're able to do now. So the goal then is to use these animals to interrogate many aspects of the cell cycle in vivo in these animals, and then also use these animals as a way to make sure when we run these preclinical trials that the drugs we're treating the mice with are targeting this phosphatase in the way our models predict it should be targeted. So I'll stop here, and I want to thank the important people that have done this work. So Yuchi Honecker is a graduate student. She's just defending her thesis, and she's been responsible for finding all of the protein kinases that regulate the turnover of CDC25A. Shirong Kai and Chris Ryan have done all the preclinical studies as well as working with the clinical samples from the breast cancer studies. Sophia Origanti is a postdoc, and she made the CDC25A reporter mice and carried out the studies in the small intestines of wild-type and P53 null mice. And then Guanhee Li is a postdoc that's leaving the lab soon, and he's the one that generated our triple knockout mice. I've mentioned our various collaborators throughout my talk, so I'll stop here and take questions. Luba?
Yeah. I'm specifically interested in the combination trials that you showed, and the fact that the effect between P53 wild type and mutant are so subtle. We have shown before that the combination of HSP90 inhibitors with a chemotherapeutic agent really requires certain timing in administration, and according to your studies as well, a genotoxic stress should be prior to inhibition of CDC25 or the Chk1 pathway. What do you think if you combine those in that sequence of giving the chemotherapeutic agents first and inducing the genotoxic stress prior to inhibition?
Right. So in the case of UCN01, the order was really, really important because it's also a CDK inhibitor. And so if you add it first, you actually lock down the cell cycle, and then you come in with DNA damage and it doesn't matter, right? And so what was really important is to add the DNA damaging agent first for UCN01. In the case of HSP90, I don't, unless it's, affecting other targets, clearly it affects a lot of other targets but Chk1, but you found the order was also important there.
That's correct.
Yeah. So, HSP90 has been shown to be important for CDK4 and cyclin D, so it may be sort of similar thing where you just lock down in G1 and then you come in with your inhibitor and doesn't matter because you can't blast through S and G2. That would be my prediction.
So in your clinical trial, did you use irinotecan prior to?
Yes, the patients get it 24 hours later than they're given UCN01. And with the mice as well. We give them irinotecan and then we come in 24 hours later and give them UCN01.
You indicated the UCN01 is a broad spectrum of kinase inhibitor. So you have also in your study in biomarker analysis, you have shown the inhibition of S6 protein phosphorylation. Okay, and then the AKT is involved in protecting cells from chemotherapeutic induced apoptosis. And how would you be able to be certain?
I'm sorry, I missed the last part of that.
AKT, for instance, it protects cells from apoptosis.
Right.
Through a bad dependent process. How would you be able to say that just because you are you come from an angle where you're looking at the CDC25, look at the cell cycle checkpoints, how would you be able to be certain that in fact any synergistic effect associated with the UCN1 could not due to something else?
Absolutely. That is a major problem with these drugs. And so I showed you the trial with AZD, which does not inhibit the AKT pathway, and we saw that similar levels of cleaved caspase 3. So but it also inhibits other things. So the question is, if you do enough of these drugs and then you say where do they overlap and where they don't overlap, and if in the end you get only Chk1 where they overlap, I think you're safer in making that conclusion.
Sort of my second question that you have for you is in your breast cancer clinical trial, would you be able to really tell whether this combination would be better than irinotecan alone?
Are better than irinotecan.
Better than irinotecan.
Yeah, that's a very good question because it's not traditionally used for breast cancer, right? So there was one study that did irinotecan alone, and so we've taken the statistics from that study and basically realized that, you know, we need an effect that's stronger than what was previously shown with irinotecan alone. And the problem with the irinotecan alone study that was previously done, they didn't stratify the breast tumor, so we didn't know what was triple negative, what wasn't. So it's a little tricky, but you're right. I think that's an issue.
Thank you.
All right, if there are no other questions, thank you very, very much.